In this video, we will discuss about bacteria-associated tumorigenesis. So we will discuss how these various different kind of bacteria affect different parts of the human beings. We will start with the stomach, where this Heliobacter pylori it interacts with this HOPQ receptors. It interacts with this CEA, CAM, which is present on the stomach using these HOPQ receptors that are present on the bacteria, and this will leads to the cytokine release or formation of cytokines in the extracellular matrix, that is CAGA, and this CAGA then will cause the activations of the target genes that helps in the proliferations, cell proliferations and chronic inflammations. So this CAGA is the main cytokines that will affect the other cells present in the stomach. Now let us talk about this large intestine. Here the bacteria that it generally affect is this enterotoxigenic Bacteroides fragilis, and in this case the bacteria that is BFT, what it will does that it will cleave these E-catherines that keep the cells stick to each other. It cleaves the E-catherines, and thereby once it enters by cleaving the E-catherines, it will enters into the cells, and that it will cause this beta catenin to disinteract. It will cause this beta catenin to be released from the E-catherines that are present in this large intestine, and this free beta catenin will then go into the nucleus, and it will lead to the upregulations of the target genes, and those target genes will thereby helps the other cells to upregulate this NANOG or SOX2 kind of genes, which helps the cells in this cell proliferations, inflammations, and also this some of the uh, stem cell transcription factors are also upregulated, which result in stemness regulations. And another bacteria that affect this large intestine is Clostridioides difficile, and what it does is that it will activate this reactive oxygen species, and once it activates this reactive oxygen, and this it will cause this NADPH oxidase that will cause this reactive oxygen species to activate. And in other case, what it will does is it will activate WNT pathway, which is the pathway for cell proliferations. And once it activates cell proliferation pathways, so it will does the same thing that is done by these other two bacteria in the stomach and large intestine. That is, it will release the beta catenin in the cytosol. It will go into the nucleus and upregulate the target genes, and this will cause the cell proliferation signaling pathways upregulated using WNT pathways, such as WNT pathways. At the same time, it will also cause ROS productions and will support the tumor cells growth, uncontrolled tumor cell growth, you can say that. And uh, in the case in other bacteria, that is PKS, Escherichia coli or E. coli bacteria, what it will does is it will enter into the cells and it will cause this DNA damage that will leads to the unique mutations. So that will affect the cells. It will affect the cells by developing the mutations within the DNA. So it will cause DNA damage, CRC specific mutational signatures. So, and some random mutations, random or specific mutations based on this CRC specific, or and these are done using this colibactin, that is when transduce or enter into the cells, which are released by this PKS Escherichia coli bacteria. Other bacteria that most of the bacteria mentioned here are about the large intestine, except the panel A bacteria, which is about the Heliobacter pylori for stomach. So in the large intestine, this Fucobacterium nucleatum what it will does is, so it will do the same thing that is done by other bacteria, but it will also interact with the sugar carbohydrate structures, that is GALNAC, and by interacting with these two structures, then only it will cause the release of the beta catenin, and it will lead to the upregulation of target genes that is related to the cell proliferations, and simultaneously it will also activate these NF-kappa beta pathways, which helps the upregulations of the pro-inflammatory cytokines. And this pro-inflammatory cytokines, thereby helps the cells to upregulate these tumor invasions uh, using this upregulation of the SOX2 kind of genes, SOX2 genes. So in total, what it will does is it will this bacteria will cause cell proliferation, cell metabolism changes, and inflammations, invasions, using this upregulation of pro-inflammatory cytokines through NF-kappa beta and SOX2 genes, and stemness regulation. So it will also, in the target genes, it will also upregulate the genes, some of the transcription factors related to the stem cells. So this is in brief how this bacteria associated tumorigenesis occurs within the stomach and large intestine. And we have discussed the mechanism of different kind of bacteria. We will discuss something related to this in our subsequent videos. So that's all for this video. If you need to understand more about their mechanisms in detail, you can go through the reference that has been provided in the description. Thank you for your time.